Okay. I guess we have most everybody, so we can get started. Just as a reminder, our review session is on Wednesday. We didn't have one today. I think a couple of students thought that the review was today. It is on Wednesday. Please make sure to accept the invite if you haven't done so already. I just want to make sure that you saw the invite, you accepted it so that I know everybody's coming to the review session. It's very important that you're there. We're going to go over questions that might be on the final. So it's good to be there and to take notes that will help you when you study and be ready. Again, I want to remind everybody that under week 15, there is a set of questions that I would like you to start working on if you haven't done so already, because again, some of these questions or a form of these questions will be on the final. And the final will be kind of similar to the midterm in terms of the types of questions. And there will be a couple of calculation questions, so you will be given enough time at the end to attach your work. So on a sheet of paper or a couple of sheets of paper, make sure you have your name and make sure to write the number of the question and the subsection of the question that you are doing the calculations. And you would type in the answer in the on Canvas in the exam itself, and the answer should match the calculations and the final answer you have on your sheet. So you will be given time to do that. So just be prepared that there will be calculations. Be prepared with a calculator as well that you're comfortable using. Um, and then on Wednesday, I will also answer any questions you have regarding any topic you wish to go over, other than just going over the questions that I have posted under week 15 for you. All right, so we're finishing up a couple of last topics here. So today I'm going to talk about food contaminants food contaminants, residues, and chemical constituents of concern. All right, so let's look here at this figure, which kind of summarizes a little bit the route of food. So you have the farm animals, crops, you have transport, you have storage, then you have processing, transport again, perhaps storage, until it reaches the final consumers. So by looking at kind of this diagram, can you either unmute yourself or in the chat box, give me examples of food contaminants, residues, chemicals of concerns that may make their way to the food and at what stage they would.
At the very beginning, you could have heavy metals in the soil of foods make it into the final product.
Perfect. Heavy metal is under chemical constituents of concern. Let's see what we get. Temperature, antibiotics, very good one. Pathogens, yes. Microorganisms, which is not going to be covered in this part, but yes, definitely microorganism. But what might come out of microorganisms that could be potentially an issue? Toxins, mycotoxins, very good. So toxins from fungus and mold, yes, for formation. So bacteria and yeah, bacteria is definitely one, but it's not a constituent chemical of concern. That you cover in microbiology. So we won't be talking on about microorganisms here. Bugs and rodents dropping in raw material. That is perfect example for the subject of our last chapter we're going to talk about on Wednesday, which is extraneous matter. So that is a separate one we call is extraneous matter. What else could be like when you are planting? And what would you potentially add to the crops? Pesticides, yes. So pesticides is a big area. Antibiotics is a big area. A lot of other chemicals, including toxins, is a big area. Heavy metal, yes. Presence of sulfite, for example, presence of compounds that form during processing, like acrylamide, or another one, furans, or can you think of others? I can think of a couple others, but I don't want to say them all. I want you to think, yeah, what would nitrogen do? It's not nitrogen per se. It is a compound that involves nitrogen for processing of meat. Nitrates, yes, nitrites and nitrates, formation of nitrosamines. Perfect. There's also, have you heard of heterocyclic amine? Have you heard of this term, heterocyclic amine? So take a piece of meat and then you are cooking that piece of meat, roasting or at high temperature, you get the meat to burn. So you have the creatine in there, and some Maillard reaction can occur, and you get these compounds that are called heterocyclic amines, and these are carcinogen. So you might not want to eat a lot of burnt meat, for instance. Melamine, have you heard of melamine? Yeah, a few years ago, it was a big deal in pet food and also in infant formula. So, melamine is a compound that is not meant for food, but it does have high level of nitrogen. So, if they formulate and add melamine, you end up, if you measure nitrogen content based on LICO, DOMAS, or KELDA, you might get higher protein content because it's based on measuring nitrogen. So they kind of boost their protein content by just adding melamine. And they didn't realize that it was really toxic substance that causes kidney failure and death ultimately. So yeah, melamine is a kind of a biological agent for adulteration, increasing the value of food with low cost ingredient. Um, yeah, the list goes on and on. There's allergens as well. For example, yeah, sulfites. Thank you, Melissa. Yes, that's a big deal for people that have sensitivity to sulfite, not necessarily the immunoreactive allergenicity, but like something similar to asthma reaction. And sulfites can be added to prevent browning, let's say in dried fruits. Sometimes it's added as a preservative, like an antimicrobial agent. Sometimes it's present naturally, so like in wine, for example. So sulfites is a big thing as well. So there's a lot. So the list goes on and on and on. But we will try to summarize some of those and give examples of detection and analysis and all of that. So annually, there are reports. They call them hazards reports. So they are reports that of contamination. And then this is an older one. In the newer book, you have newer references and percentages. But anyway, in here you have a distribution in terms of how much is how much are chemical hazards versus mycotoxin versus microbial versus other. So you can see them, like the chemical is this big part of the chart, mycotoxin toxins, microbial, and then others. So under chemicals, we talked about heavy metal is considered chemical, pesticide is considered chemical, antibiotics, other hazards like biological hazard and fraud, like melamine, for example. Labeling is another issue. Bisphenol A, for example, I don't know if you guys heard about it. It leaks from the packaging material, and there were a couple, like a case a few years ago about the plastic bottles. You have bisphenol A leaking from the bottle into the water or the juice. Also, sometimes the ink material can seep from the container into the material. For example, cereal. Now you see your cereals mostly are in a plastic bag inside of the box, because to prevent any ink from the box reaching to the cereal. So yeah, a lot of different ways these contaminants can make their way to the food, or they can be present naturally in the food, or they can be part of present due to processing. So it's good to know what we have and how we can prevent them from being there at the level that we call above the tolerance level. So there is a tolerance level for these different compounds, and it varies. Sometimes there is zero tolerance level. For example, aflatoxin B, there's no tolerance level for it. It is genotoxic, not just carcinogen. So it is very toxic, so there is no tolerance level for it. So the EPA, so the Environment and Protection Agency, sets these levels. So that agency basically determine what would be a tolerance level, and then the FDA and the USDA, they enforce it, making sure that everybody follows that. So big categories are pesticides, antibiotics, mycotoxin, allergens. These are big categories, so we'll talk about each of these categories separately. And then chemical additive, there's a whole slew of them, adulteration or food additives, and many others that form during processing, for example. So we'll talk about all of those. So there are a couple of very informative tables in the book that would be good to study from. So the first one here is table, let's see, 33.1. And this table summarizes main contaminants, pesticide, mycotoxin, antibiotics. GMO is another area we didn't talk about, allergens, sulfites, and nitrite. So here it summarizes some quantitative methods of analysis, and then a semi-quantitative method of analysis. It's good to study from and remember a few examples for each of these categories. What would be a quantitative method? What would be a semi-quantitative method? There is another table that is important. I'll show you that later. So at first, there are general consideration, and this has to do with sampling. If you remember from the beginning, we talked about sampling in the beginning of the semester. So sampling is very important, especially when you have something that you're looking for that is not normally distributed. We're not looking at total moisture or protein or fat. We're looking at something might be there on one corner of the food product and might not be there in another. So it's kind of randomly distributed, and you want to do something we call attribute sampling. So you got to remember that. That's why the final is cumulative, because there's certain things you really have to remember and know. It carries with other chapters. For example, here it's important to remember your attribute sampling. With attribute sampling, you need to make sure you have a representative sample, and you need to take a lot of samples. Because if you let a sample pass, a whole production pass, because you didn't sample enough, that's a hazard to the consumer. So you want to make sure that when it comes to something that could be hazardous to the consumer, you do attribute sampling. That means you take a lot of samples from different places, you homogenize, and sampling error in this case is more impactful than analysis error because, again, it has an impact on the consumer. So you do not want to sell product that could potentially make people sick. All right, so basically a very important part is to have a representative sample, have a lot of samples taken out, and the sample is homogeneous for sampling. You collect, you store under proper storage condition, and then when you're ready, you start your sample preparation. So really here you see that sample preparation starts with the crude extraction. So first of all, is it a water soluble? Is it organic soluble compound? So you start with extracting your constituents of concern based on what you know about them. And then after you extract them, you want to clean up the fraction. You want to remove from the crude extraction components that might interfere in the assay. So you do cleanup. Oftentimes it's a chromatographic cleanup that you would do, and you concentrate because some of these components are present at very, very, very small concentration. You want to enhance the sensitivity of the assay, so there is a concentration step that you would do. And then if you're doing a GC, let's say analysis, and your sample is not volatile, is not stable, thermally stable, there would be a derivatization step. So it's not always necessary. It depends on the method of analysis. Then in most cases, it's a chromatographic analysis. You see that we have a few immunoassay-based analysis, but a lot of the time it's chromatography coupled with mass spectrometry. So oftentimes GC or HPLC with MS as means of detection. So one thing I want to say on the note of means of detection, if you see in the exam a question asking about method of analysis and mode of detection, that means I'm asking you, are you using chromatographic method? Which chromatographic method? And you would explain that, or an immunoassay, okay, you would say it's an immunoassay. But when I say, what's the mode of detection, then is it a mass spectrometer? Is it a UV detection, a spectrophotometer? So the mode of detection is how we are detecting the components. So there's the method, and then there's the detection. So after separation, we have the identification and quantification. Sometimes you just want to get a qualitative answer, whether they're present or not. Sometimes you want to quantitate, and then you report. So the choice of the extraction and the choice of the separation depends on what do we know about the sample. So the food matrix, how complex is the food matrix? What are the interfering components? What do we know about the analyte? Is it polar? Is it non-polar? Is it volatile? Is it thermally stable? What do we know about it? What's the molecular weight? Does it have nitrogen? Does it have sulfur in it? So a little bit about the properties of the analyte. Feasibility, what do we have in the lab? There could be several methods, but then you have to go with whatever you have available. And then multi-residue versus single residue. Sometimes you really want to purify a single residue and determine tolerance level. If you're doing that for a pesticide, for example, your extraction is only targeting one component, not 5 or 6 pesticides. The whole method is targeting one component. Multi-residue, if you know you have pesticides that are similar in molecular structure and you can analyze them just to know if they're there and how much they're there to determine their quantity, basically you can do a multi-residue analysis. So these are the basic steps. So sample preparation, removal of edible and non-edible fractions. If we have a peel, we remove the peel, we remove the pits. Homogenization is very important. Then it's often solvent extraction and filtration, and then the cleanup. You can do cleanup using different methods, chromatography based on size exclusion or adsorption or even affinity. If we're really trying to purify one component, oftentimes affinity chromatography would be good. Many times we use solid phase extraction. So this specific term is used. So if we look at the letters, so what does that mean? Q, U is for quick, E for easy, CH for cheap, E for efficient, R for rugged, I mean also kind of form of efficient and quick, and S is safe. So this is a very common method of cleanup. So it can be regular solid phase extraction or solid phase micro extraction on a micro scale, depending on the concentration of the samples. And the accelerated solvent extraction, we talked about it during the fat analysis chapter. And then the analysis, after you extract, you purify, you run the analysis GC, HPLC, thin layer chromatography. If it is qualitative, you just want to screen presence of or not. And derivatization, if we need to use GC, but we need to derivatize to enhance thermal stability and volatility. Mostly MS identification, and a lot of the time we can also use immunoassays. So rugged, I think there's a question about what the R stands for. Rugged is like when you say a workhorse, it means like you have good throughput. It's just, you know, it's a good solid method with high throughput. Yeah. Okay. Okay. So let's go with pesticides now. There's so many pesticides out there. The list is humongous. The last time I checked, there were like 1,300 registered pesticides. I'm sure there are more now. So, but in order for the pesticides to be used, they have to be registered, meaning they have to be tested for safety. They have to have a tolerance level, toxicity known. So they have to be well thoroughly studied before they're approved for use. And oftentimes, like I said, we do multi-residue analytical testing. You want to screen so many different pesticides that may belong to the similar family or multiple families, but similar in structure. So you extract and run one chromatographic method, and you're able to see few and determine the concentration of few in one method. Like I said, single residue methods are not commonly used unless we are really trying to characterize the pesticides and determine its tolerance level. You can do that with immunoassays. We're using antibodies very specific to this pesticide. And there are methods that are unique in terms of the extraction and then utilizing specific column, either GC or HPLC for measurement. Semi-quantitative and qualitative. So this is done with thin layer chromatography and enzyme inhibition. And I'll show you example here in a little bit. So this is a multi-residue assessment of few pesticides. And here, if they are non-polar pesticides and volatile, then they can be done using GC. And then the detector can be either flame photometer or electron capture, for example. It really depends on the components of your analyte. So if you have pesticides with sulfur or potassium in there, you can use flame photometer as a detector. A lot of times, really, you rely on mass spectrometry as a detection. We definitely would be great to have internal standards as well. That would enhance the accuracy of your detection. Sometimes the pesticides are polar, and a lot of the newer pesticides are polar pesticides. So instead of going through derivatization and running GC, then you would develop HPLC methods. And there are a lot of HPLC methods that are used with polar pesticides. And then the detector is really based on the analyte. Does it absorb light? We can use UV detector. If it doesn't have any specific characteristic, we can use the universal one, which is the light scattering detector. Or we can use mass MS, which is also the most common one used for pesticide analysis. Now, would you use LCMS or LCMS-MS? So with MS, great, you get a very nice spectra that is unique to the compound. However, if you have MS-MS, you get the cleaner spectra, because you take out some of the noise and you take that one fraction and you send it to another MS, you fragment it more, and then you get smaller fragments, then you can get better structural identification. So you can compare your standard here becomes very close to the one that is present in the sample. So usually LCMS-MS gives you more fragmentation data and more information about the structure so that you can have more confidence in identification. Immunoassays, since they are small, pesticides are small in molecular weight, then you have to remember we use competitive ELISA. You do not use sandwich ELISA or indirect ELISA. Does not work for small compounds. You have to do the hapten, which we talked about last Friday. We can do screening by thin layer chromatography, especially for pesticides that actually inhibit a specific enzyme in insects or pests, which is the cholinesterases. So if you have pesticides that inhibit this enzyme, then what you would do is you would extract your sample, you run on each lane, you run different samples, and then you spray the plate with the enzyme, with this cholinesterase enzyme. And after you spray it with enzyme, you spray it with the substrate. And the places where you don't see a color, because enzyme and substrate, you generate a color. The places that you don't see a color, that means you have pesticides. And the pesticides inhibited the enzyme. So you don't see a color, the substrate does not change into a product. So you don't see a color. Okay, moving into mycotoxins, need to make sure I keep track of the time. Okay, moving into mycotoxins, these are compounds generated by fungus. Mostly Aspergillus flavus is one of them, but there are other fungus that obviously that produces toxins. Most of the toxins that are of concerns are aflatoxins and ochratoxins. And there are different types of aflatoxins, A and B, different Bs even. Aflatoxin B1, for example, is the one that I told you earlier. Is a geneticide. That means it's not just carcinogen, it impacts DNA, and then there's no level of tolerance for that particular one. So they can be present in commodities. Sometimes you don't see the fungus, but it could be there. Sometimes you have fungus, but it's not necessarily there. So if there is any chance that it could be there, there has to be testing and not relying on visible fungus presence or not. In most cases, tolerance levels are really, really low, less than 15 parts per billion. Methods of analysis for mycotoxins are mostly, if you want to do quantification, it's mostly reverse phase HPLC coupled with UV detection or fluorescence or MS detection. Thin layer chromatography, you can purify with immunoaffinity if you want to purify them and uh, determine their identity and to quantify exactly a certain mycotoxin. And you can do immunoassays similar as we talked about last time, which is the comparative ELISA. Another category is the antibiotics. What are the drug residues? They are added to treat a sick animal, so that we call it therapeutic levels, but oftentimes they are used to enhance the growth, to increase the weight of the animals. So they're added at subtherapeutic level. So that to increase weight gain and also reduce chances of infection. But if the animal is meant for slaughter, and meat use, they have to stop a few days or a couple of weeks, even I'm not sure, but they stop the antibiotic prior so that it's clean from the system because there's zero tolerance level for antibiotics. The same if the animal is going to go through lactation and collection of milk, there should be a phase where they don't have any antibiotic in their system. So there is no tolerance level for antibiotic because people have allergies to antibiotics potentially. And also if you continue using certain antibiotics, you get bacterial resistance. And if you have milk, let's say with some antibiotic, it will impact processing if we're producing fermented yogurt or if we're ripening the cheese, if there are antibiotics in there, that's going to impact that whole process that require microorganisms. So qualitative methods, screening methods, turbidity measurement. So you would have your sample and you add a media and you add a certain microorganism and then you watch for growth of that microorganism. The more turbidity you get, that means you have more growth. Another method is the disc diffusion method. So you see here the little discs here that you have. So you have your agar or culture material. So you put those little discs and inside of these discs could be your sample, let's say milk sample. So you have a little bit of milk sample in each one of those. Then you inoculate with a particular bacteria, for example, Bacillus stereothermophilus, and then you incubate. If there are zones of clearance around that disc, that means this sample had antibiotic that leaked out of that disc and prevented the growth around this area. So this is kind of just, is it there or not there? You don't do quantification here very much. Charm test is more for quantification. It's very common for antibiotics. In there, it's a competition between an antibiotic that is radio-labeled and then the sample that potentially contain antibiotic. So you add bacteria, and then the antibiotic would want to interact with the bacteria. If you have a lot of antibiotics in your sample, then they would react with the bacteria and then there would be less radioactivity. If you don't have antibiotic in your sample, or a little of it, you'll see more of the radio-labeled antibiotic attached to the bacteria, so you have higher radioactivity. So of course you need a standard curve in order to quantitate based on the radioactivity reading. So quantitative determination, again, reverse phase HPLC, you can measure UV detection or you can do GCMS. Allergens. So allergens are important given the fact that we have about 90% of food allergy coming from these big 8 allergens. So milk, eggs, fish, shellfish, tree nuts, peanuts, wheat, and soybean. The list is going to grow pretty soon. Soon we're going to have a big 9, big 10 with more proteins incorporated in our foods. The more we're going to see allergies. But for now, we have these allergies, and you know that you have an allergen in the food product if you have a label contains soybean or contain peanuts or eggs and so on. This is due to the Food Allergen Labeling and Consumer Protection Act, where we have to list the ingredients by their common name so that people can read and recognize, oh, it has wheat, so it contains gluten, or it has eggs, or whatever. You cannot use scientific names for ingredients. It has to be known by their common name. And also, if it's manufactured in a plant that also manufacture allergens, it has to be on the label. For example, if you have M&Ms, if you read on the package of M&M, they were produced where peanuts could have been there, so there could be a peanut contamination. So it has to be on the label. So how do we test for those? So we have the protein methods and we have the DNA methods. So each has their advantages and disadvantages. Sometimes you have a food product that contains a protein ingredient, like soy protein, potentially at a very low concentration, but it has soy protein, or it may contain dairy protein, whey protein, isolate, but at a very low concentration. And you want to be safe and you want to test for that. If you use the DNA method, which is based on PCR, you might not detect presence of soy or whey. Why? Because you're working with a protein ingredient that the way that you isolated that protein ingredient to add it in your food, you must have lost the DNA component. So you might not detect their presence if you just use DNA. The protein methods would be good methods, but also we have some limitations there. This image here, which is figure 33.5 in your book, it shows an extract from peanut. And one of the most allergenic protein components of peanut is ARH something, ARH something. And this is here and here where they're circled. So in image A, you can see them with the Western blot. In image B, you don't see them. So the difference between A and B is the protein was extracted differently. The protein in image A was extracted with some salt in the extract, in the buffer extract or extracting buffer. Whereas in B, the buffer did not contain salt. So these proteins were not present. So you might wrongfully deduce that you don't have these allergens in this ingredient. So again, that's something to watch for with protein-based assays, is how we're extracting the protein for analysis. Also, processing impact interaction with the antibody. So if you have specific antibodies in an assay that you purchased, already designed assay for specific proteins, but the proteins have been formulated in a food in such a manner that the structure changed, is different. When that happens, and then you're trying to test them using an ELISA kit, the ELISA kit may not detect them, but are they not allergenic? They are still. They could be still allergenic because other antibodies in our body could react with them still. But the assay that we use with specific antibodies, monoclonal antibodies, won't detect them. So that is a problem too. So we have to be really very careful in which method to assess and be aware of the advantages and disadvantages of each. Now, moving on to the general list, a lot of them, right? So we talked about the main ones, the pesticides, the antibiotics, the allergens, the toxins. All right, but the list is huge for other chemicals of concern. Sulfites and nitrites, melamine, packaging material, furans and acrylamide due to processing, benzene that could naturally be present, monochloropropane that could be the results of the production of texturized vegetable protein or potentially protein hydrolysis. Perchlorate also could be present in the soil, in water contaminated from fuel, and many others here. So what we'll do is we'll go over a few of those. But the methods of analysis are currently expensive. They're not rapid and mostly involve chromatography and mass spectrometry. And limit of detection is important because some of these, their tolerance level is very, very low. So we have to have methods that be able to determine the quantity at a very low concentration. There's always research into looking at faster ways and more effective ways and less expensive ways to analyze these compounds. Looking at FTIR, for example, as a method for quick quantifications, or IR in general. So looking at sulfites, like I said, sulfites, they are used, either they're added to food or occur naturally, and there are different forms of them. There's so many different forms: sulfur dioxide, sulfurous acid, inorganic sulfite salts. The list is huge, and sometimes the methods does not necessarily measure all of the form. Some methods measure most of the forms, some methods are specific for specific forms. So they cause some sensitivity reaction, asthma-like in some folks, and it is required to put on the label whether or not, well, if they have any sulfite level above 10 parts per million, you should put on the label contains sulfite. So there are different types of method. The most common method is Monnier-Williams. So if you want to remember any method, remember Monnier-Williams for sulfite. So it's basically measure most of the sulfite in the form of SO2. So sulfite that are free and plus some sulfites that are bound. It is the method that the FDA recommend for labeling. So that's the method I say, try to remember that method. So basically it's based on the principle of heating with HCl. Then you convert all of your sulfites into SO2. Use nitrogen to sweep the SO2 and reacted with hydrogen peroxide. SO2 reacting with hydrogen peroxide will give you sulfuric acid, and the sulfuric acid is measured either by measuring turbidity or measuring the weight. So remember this principle, easy. Heat with HCl, produce SO2, react SO2 with H2O, produce sulfur hydroxide, or sulfuric acid, sorry, and then you measure by turbidity or by measuring the weight gravimetrically. There are other methods. There's the Ripper method, which is commonly used in the wine industry. It measures free SO2. This is specific. It is based on a titration. Enzymatic method. This is another method that measures sulfites and utilize NADH peroxidase enzyme. HPLC coupled with fluorescence is another method as well. So nitrites and nitrate, they are used as preservatives and used to cure meat products, give you that very characteristic pink color. It prevents browning. It's added sometimes to dried fruits. And also it acts as a preservative against bacteria, specifically Clostridium botulinum here in this case. It can be sometimes present naturally, like in celery. There are legal limits for both, and the health concerns is that production of nitrosamine, which is a suspected carcinogen, which is mostly produced upon high heat treatment. So colorimetric method of analysis for nitrites, and you can also use chromatographic methods. So you don't have to remember really the details of the colorimetric method. Just make sure you remember there are two AOAC methods. One is based on colorimetric detection and one is chromatography. So there are rapid methods, the ion selective electrodes we talked about last week in the salts or in the mineral chapter. There are electrodes specific for nitrates and nitrite, that's very common, and also test strips for nitrite nitrates as well. Now, this is the table that I would say would be good to study from if you were given a question with examples. I saw a question that I missed. Is the charm test only used for milk sample? No, it's not necessarily only for milk sample. It can be used for other samples. I gave milk as an example. Okay, so this table here, it lists a lot of different compounds of interest: acrylamide, benzene, bisphenol, etc. So it gives you the nature of the enzyme, nature of the compound, and why it made it to food. For example, acrylamide is formed in products that are high in carbohydrate and got high temperature treatment. So, for example, frying of potato chips, and you have asparagine as one of the amino acids in a protein, can react with reducing sugar, and then you get acrylamide. So it would be good to know how it made it to the food. Why is it of concern? Carcinogen, for example, a sample example of a food, and a method. So it would be good to study. This is a good study table. I can tell you if there is an exam, a question in an exam, I might ask you why this compound is present in this food, or give me a food example where that compound can be present. What would be a method of analysis? And why is it of a concern? So these are could be a potential component of a question. All right, we'll talk about each one of them separately next time. So I ran out of time. I just looked at the time. This will take 5-10 minutes to finish. So what I'll try to do so that you know before you leave right now is on Wednesday morning, 9:30, when we start our review session, I'll spend about 20 minutes, 30 minutes finishing up this lecture, and then the extraneous matter usually takes me 20 minutes or so to finish. This way we would have covered all the material, and then we'll start the review questions. Okay, so I'll see you guys on Wednesday. If you haven't accepted, please do so that I know that all of you are coming. All right, see you later.